All right, welcome back. Let's continue from where we left off. And where we did leave off was after we completed the absorption of amino acids, right? Right. So now, without further ado, I think it would only do justice if we start the real metabolism of amino acids. All right. However, before we start the real metabolism of amino acids, let me ascertain you that first we study the general metabolism of amino acids. All right. So first, the general metabolism of amino acids that we take care of, and that itself is a bit of a long topic, but it's easy enough. After we complete that, the general part, we go on to the specifics of the metabolism of the different amino acids. So then we will be looking at the individual amino acids and their metabolism. All right. But for now, we focus on the general. Is it visible? Oh yeah, it is. Okay, so the general metabolism of what exactly? General metabolism of amino acids. Right. We talked about the digestion, absorption of amino acids. So are they not the part of metabolism? No, they are not. And frankly speaking, I did not talk about the digestion of amino acids. Remember the first favor you did me in the last video? Yeah, by going through digestion yourselves. It's not that important. We have been studying that for a long time now. So digestion, absorption, are these parts of the metabolism? That's the question. Answer is actually no. Metabolism is kind of different from digestion. How? You see, metabolism is how our body utilizes these amino acids. Like we are talking about the metabolism of amino acids here, so that is why I'm taking the example of amino acids. Metabolism is how our body utilizes amino acids, right? It is a sum total of all the chemical reactions, of all the chemical processes that are going on in the body. And since we're talking about the amino acids as such, so all the chemical reactions taking place in the body pertaining to the amino acids and how our body is utilizing the amino acids. That may be for different purposes, obviously. It might be for building something, it might be for providing energy, whatever. Those are the specifics. We will be dealing with those later. But for now, we need to understand the basic concept, right? That is metabolism. Now, what is digestion? Digestion is how we are getting the amino acids itself. Are we in our diet taking amino acids directly? Do you have a cup of alanine and glycine in the morning when you wake up? Obviously, no. You don't take a cup of glycine and alanine like that, right? You eat a protein rich diet. It has protein. Proteins have amino acids as their building blocks. But you're not actually consuming amino acids directly, right? You are actually consuming amino acids in the form of proteins, peptides. So how the body is actually breaking those down and the amino acids are being formed in the body, that is the story of digestion. How these amino acids are being taken up from the digestive tract to the different parts of the body is the part of absorption and transportation. Now, the story continues in a new chapter, which is general metabolism of amino acids. This is essentially the new chapter in our story of amino acids. And frankly speaking, this is what the chapter is about, the metabolism of amino acids. Now, here's the thing. Amino acids, what do they contain? What do they contain? What do amino acids contain? Amino acids contain this group, if I'm not mistaken. Am I mistaken? No, I'm not. They must contain this group. This is the amino group. Right? So they must contain this amino group. Right? Right. Now, if we are talking about metabolism of amino acids, so we have to talk about what happens to this group. What happens to this amino group? Because amino group is a basic constituent of all amino acids. This is the identification of amino acids. They contain this, right? Along with a C end as well, a carboxyl end as well. Hence, they are amino acids. So, what happens to this group during the metabolism of amino acids? That is a question we are asking ourselves here. The answer is very, very, very important. All right? The answer is this being formed, ammonia. So, we can say that the formation of ammonia is an extremely important step in the general metabolism of amino acids. Am I clear up to here? I hope I am. Very well. So we concluded that ammonia has got to be formed in the metabolism of amino acids. Right? Right. How is it formed? Why is it formed? What happens after it is formed? This is what we will be talking about. All right? Right. So suppose we have an amino acid, right? We have an amino acid. Where is this amino acid present? This amino acid is present in the cells. In the cells. Was it always present in the cell? Of course not. We took it with our diet. All right? It was in the digestive tract. It got digested. The peptides got digested. This was formed. This was absorbed. Now we have it in the cells. Now the metabolism starts. All right? Now the metabolism starts. Pertaining to the metabolism of amino acids, the first thing that takes place is a process. A process that is known as transamination. Transamination. This is the process that takes place with the amino acids, or rather, yeah, I'm right, with the amino acids after they have been absorbed, and this is pertaining to the metabolism of amino acids. What is transamination? Let's talk about that. If we are to define transamination, we would say that it is a reversible transfer of the amino group. Yes. So it is a reversible transfer of amino group. From where? From obviously an amino acid. To where? This is an important thing. From, I said, from an amino acid, right, to what we say to be an alpha keto acid. Yes. Now we come to a very, very important thing, that is the alpha keto acid. What is this happening? Let me give an example and this would perhaps be clearer. I hope so. What's happening is, just as I said in the definition, that it is a reversible transfer of an amino group from an amino acid to an alpha keto acid. Alright? So suppose we have an amino acid here. And suppose this is amino acid 1. Alright? This is amino acid 1. What happens is, this reacts with an alpha keto acid. This reacts with an alpha keto acid. Now, we say that this alpha keto acid is at the 2. Alright? This reaction, what does this give us? This gives us amino acid 1 transfers its amino group, just like I defined transamination, right? We are actually talking about transamination here. So amino acid 1 transfers its amino group to this alpha keto acid, forming an other amino acid. This is the second amino acid. And itself gets converted to an alpha keto acid. Alright? So amino acid 1, after its transfer of the amino group to the alpha keto acid to the alpha keto acid, forms an alpha keto acid. Alright. I hope I did not puzzle you guys. Amino acid transfers its amino group and forms an alpha keto acid. To whom does it transfer this amino group? To an alpha keto acid, hence forming an amino acid 2. So it's just reversed. It's kind of just reversed. Yes. The amino acid gets converted to an alpha keto acid gets converted to an amino acid. This exchange, this jumbling up is known as transamination, right? Now, this is a chemical reaction. What do chemical reactions need? Chemical reactions need a catalyst. They need a catalyst, right? So this is a biochemical reaction that is going on. So biochemical reaction needs a biochemical catalyst or an enzyme. So this reaction is no exception to that. And the enzyme which is used, since this arrow is too small to write the enzyme name over it, I'm going to write it over here. And the name of the enzyme is, or rather it's not the name of the enzyme, it's a group of enzymes. And what are they named as? They are named as amino transferase. Amino transferase. They are also known as, transaminase. Alright, so aminotransferase or transaminase is the name of the group of enzymes which catalyze this transamination reaction. Alright, now let me tell you something. So this is what happens in transamination reaction. Alright, why am I talking about transamination reaction again? Because this is the first step in the metabolism of amino acids. An amino acid, after being absorbed, pertaining to its metabolism, this is what happens first, that is transamination of that amino acid. Now, there are obviously exceptions to this. There are some amino acids which are not transaminated, but we will talk about them later. For now, let's follow the general trend and understand the things first. These aminotransferases or transaminases, as I said, these enzymes, they need what we say to be pyridoxal phosphate. Pyridoxal phosphate. All right. What is pyridoxal phosphate in this case? We have kind of learnt enzymes, so we should know that this is a prosthetic group. All right. Prosthetic group. So what is the prosthetic group? Pyridoxal phosphate, or PLP. Pyridoxal phosphate. It is used as the prosthetic group in these enzymes. These two enzymes are not different. They are the same name given to the same group of enzymes, that is aminotransferases or transaminases. Same thing. Right. Now, I said that this is a reversible transfer. So, just like this way can happen, this way can also happen. All right. So, this can also take place, this can also take place. And this has its own utility. We will be talking about that in just a minute. Now, I said something about these enzymes having pyridoxal phosphate as their prosthetic group, right? So, pyridoxal phosphate, how exactly is it attached to these enzymes? Like, what is the bond there? What kind of a linkage is present in there? Now, this goes on to what I had discussed in the previous video about this molecular thing. The molecular thing is something which is going on parallelly in biochemistry, obviously, because biochemistry means there is as much chemistry as there is bio in it. But we, being in a profession which does not really need the molecular things or the chemistry part that much, so we kind of ignored that. But here we can make a short note as to say that the bond or the linkage which holds this prosthetic group pyridoxal phosphate to the enzyme is a skiff base link. A skiff base linkage. All right. That is the link which is present, that is the bond which is present between the pyridoxal phosphate and the aminotransferase. It's not that I don't feel like that is very important pertaining to clinical application, because that is after all what we will be aiming for to. But still, just to make a mental note of this. Alright, this is the first favor that you're doing me in this video, that you just remember the skiff base linkage. Right. So that is transamination reaction. Now, let's give some examples of transamination reaction, right? We just said about amino acid 1 and amino acid 2. But let's be a little more specific and see how this actually works in the real world. In the real world, I mean the real case amino acids, all right? So let's take an example. The example that we take is suppose it is alanine. I was talking about alanine, right? So alan. All right. Alanine. All right. Very good. So alanine is an amino acid, and we are talking about the transamination reaction of alanine. So alanine is amino acid 1 here. Alanine is amino acid 1 here. Okay. Okay. What is the alpha keto acid that we will be using here? Now, I want to make a point very, very clear that the alpha keto acid that we use here, as well as for most transamination reactions, is alpha keto glutarate. Very, very important. Alpha keto glutarate. All right. Alanine plus alpha ketoglutarate. What will it give us? What will it give us? Alright, it's a bit narrowed down, so reversible sign. I know this is kind of strange, but we have to improvise here. Anyways, the thing that is happening here is alanine will be converted into the corresponding alpha keto acid, and alpha ketoglutarate will be converted to its corresponding amino acid. Why? Because alanine is actually transferring, although reversibly, its amino group to the alpha ketoglutarate. So what is alpha ketoglutarate forming? An amino acid. And what amino acid is it? Well, the amino acid it forms is glutamate. Glutamate. You guys remember glutamate from the previous video? Of course you do. Glutathione. So one of the constituents was glutamic acid. Gamma glutamyl cystinyl glycine. So glutamate was one of the constituents there. So alpha ketoglutarate, after being transaminated, forms glutamate. And this is a very important amino acid. Plus, alanine forms its corresponding alpha keto acid. What is that? What is the alpha keto acid corresponding to alanine? That is a very, very common compound, pyruvate. Pyruvate. Obviously, we are using an enzyme here, and obviously that is PLP related. What is the name of the enzyme here? The name of the enzyme is alanine aminotransferase. Oops, sorry. Alanine aminotransferase. When abbreviated, this gives ALT. Now I want to make a note here. If you remember, although obviously I have not taught that, but ALT is a very, very common name as far as biochemical tests are concerned. When we do a liver profile means we are actually analyzing the function of the liver. So we pertain to a liver profile there, and the different enzymes that we actually kind of quantitatively measure from the liver include alanine aminotransferase, ALT. ALT has another name, that is SGPT. SGPT. Alright. I implore you to remember these two abbreviations. If you remember these two abbreviations, you will be able to remember this reaction as well. Because why I'm telling you this is because it might be confusing as to what was the product which was formed when alanine is transaminated. We will see later that alpha ketoglutarate and glutamate form the obligatory alpha keto acid and amino acid pair. Do you get me? Alpha keto acid, amino acid, this is a pair. Amino acid, alpha keto acid, this is another pair, right? This alpha ketoglutarate, if we replace alpha keto acid 2 with alpha ketoglutarate, the amino acid 2 will be glutamate. This pair here, these two are obligatory, almost compulsory. Like I said before, a few minutes earlier, that most transamination reactions use alpha ketoglutarate and glutamate. That is why this glutamate is so important amino acid. Alright? Because most, like I said, most transamination reactions are taking place like this. So we will obviously remember these two. But what happens to alanine and pyruvate? We have to remember these two also. For remembering them, that is why I implore you to please remember ALT and SGPT, these two abbreviations, because these two enzymes, or rather, this is actually the two different names of the same enzyme. ALT stands for alanine aminotransferase, and SGPT, this has a big name. S stands for serum. So you can actually exclude the S part, that is the serum part, because we say SGPT when we are actually extracting this enzyme from the serum. That is why serum comes here at the first. You may exclude this when we are talking about transamination reaction. You may exclude this S. You can say just GPT. So what does GPT stand for? Glutamate pyruvate transaminase. Glutamate pyruvate transaminase. So serum glutamate pyruvate transaminase. This is the name. And ALT stands for alanine aminotransferase. These two are the same names given to the same enzyme. Alright? Right. So if we remember these two abbreviations, we will be able to pin down all these four things. See, if you remember ALT, alanine aminotransferase, you will remember that you're talking about alanine as an amino acid. So alanine, alright? If you remember GPT, you will remember that you have a glutamate. So you will obviously have an alpha ketoglutarate here. And you will remember that you have a P, that is pyruvate. So pyruvate, alright? This is the thing. Obviously, this would require pyridoxal phosphate, PLP, as a prosthetic group. Right. Right? Right. Now, let's just take another very important example very quickly here. That is, we talked about the enzyme, or not enzyme, the amino acid alanine, right? Right. Now, suppose we take the example of a different enzyme, that is aspartate. Aspartate. So aspartate, when that reacts with, what it would react with? What did I say was the obligatory alpha keto acid? Compulsory alpha keto acid? Yes, alpha ketoglutarate. Alpha keto glutarate. Alright? Alright. Aspartate plus alpha ketoglutarate would give, obviously, let's write the known things first. That is glutamate. Alpha ketoglutarate will give glutamate. We know this. Plus aspartate will be transaminated to a beautiful product, oxaloacetic acid. Oxaloacetic acid. Again, the enzyme used will be AST or GOT. AST stands for aspartate aminotransferase. GOT stands for Game of Thrones. Oh no, I'm sorry. GOT stands for glutamate oxaloacetate transaminase. So GOT does not stand for Game of Thrones here. GOT stands for glutamate oxaloacetate transaminase. See, if you remember, that is what I was talking about just a few minutes ago, that if you remember only the enzyme abbreviations, these two names, you will be able to pin down the reactants and the products. Although this is a reversible reaction, so there is no such thing as a reactant and a product, but still, you're getting the general picture here. Obviously, we are using the pyridoxal phosphate here. So there is what we say to be a reversible transfer of amino group from the aspartate to alpha ketoglutarate. Aspartate hence forms oxaloacetic acid, which is its corresponding keto acid, and alpha ketoglutarate hence forms glutamate, which is its corresponding amino acid. This is transamination. This is transamination, and this is exactly what happens with amino acids when they are first absorbed. So this is the first step that actually occurs pertaining to the metabolism or pertaining to the general metabolism of amino acids. So what is the significance of transamination? Is it only the fact that it is the first step of a general metabolism? Well, of course. Of course, it is the first step of the catabolic metabolism of amino acids, and hence it is special importance. But it has another very important importance. Importance. My God, what an amazing thing I've said. Importance, a very significant importance, lies in the fact that this reaction is reversible. Are you getting what I'm talking about? You see, amino acids can be classified into essential and non-essential, right? Essential amino acids are essential for the body, and non-essential amino acids are not essential for the body, right? No, of course not. They're both essential for the body. Then why is this naming like this? It's because essential amino acids are essential in the diet. These amino acids, that is essential amino acids, they are so essential that they cannot be synthesized by the body. So they are essential because we need to obtain them from an external source, that is our external diet. Non-essential, on the other hand, we can synthesize that inside our own body. Non-essential amino acids is where transamination comes. Alright? Non-essential amino acids, how can they be synthesized? See, aspartate is a non-essential amino acid. Why? Because when you reverse this reaction, you will be forming aspartate. That's true, right? That's very true. Suppose we need to form aspartate. Rather than degrade it, rather than metabolize aspartate, we are at a lack of aspartate in our body. So our body needs to synthesize aspartate. So this reaction can be reversed, because I already said that this is readily reversible reaction. So to form aspartate, we can readily reverse this reaction, glutamate and oxaloacetic acid. Same transamination reaction will take place, same enzyme, same prosthetic group. These will be given. So aspartate is a non-essential amino acid, and non-essential amino acids can indeed be synthesized by this process of transamination. Same goes for the previous example of alanine. Alanine is also a non-essential amino acid because it can be synthesized in the body if we reverse the reaction. Alright? So synthesis of non-essential amino acids is very important function of transamination. Alright? Now, do you remember that I said about exceptions to this? There are some amino acids which are not transaminated, as such. What are those amino acids? What are those amino acids? There is a kind of a mnemonic, I would say. I kind of formed it. Lie 3 times and you are a pro. Lie 3 times and you are a pro. If you lie 3 times, you will become a pro at lying. Alright? Makes sense, and making sense is all we need. Lie stands for, okay, if I say lie, which amino acid do you think I'm talking about? Correct. Correct. Very good. Lysine. Right? 3. If I talk about 3, which amino acid am I talking about? 3. Yes, threonine. Very good. And if I'm talking about pro, which amino acid do you think I'm talking about? Very good. Prolin. Beautifully said. Lie 3 times and you are a pro. So what do these intermediate words mean? These words are only there to make the sense in the sentence. All right. You can also remember this by another hint, that is, you see, 3 means that the number of amino acids will be 3. Am I making sense here? You already have a 3 in the sentence, right? That means firstly there are 3 examples and 3 is also acting as threonine. All right, I think this is a multi-purpose mnemonic if you ask me. Okay, anyways, where is this transamination taking place? Where is this taking place? Inside the cells, because we are talking about metabolism, so this is obviously inside the cells that we are talking about. But to be more specific, where inside the cell are we talking about? Where inside the cell actually? Mainly in the, what we say to be, cytosol. All right. All cells? Are we talking of all cells? Yes, kind of. Cytosol of almost all cells. So transamination takes place in the cytosol of almost all cells in our body. That is how important this process is. All right, that it takes place in the cytosol of almost all cells. All right, transamination is forming this glutamate, because there is an obligatory thing that is being formed here, and another amino and another alpha keto acid, not amino acid, another alpha keto acid. All right, now what happens? Is this the end? Of course not. This is just the beginning. Now another process takes place. Another process takes place. This process is known as what we say to be oxidative my god, it took a long time to write that. Oxidative deamination. Oxidative deamination. I don't know whether any of you guys have watched a web series known as Breaking Bad. Have you watched it? Or are you guys too nerdy to watch that? No, it's a very good web series, and it's about a chemist actually, and the different choices that he makes in his life. So why I'm talking about Breaking Bad is that in Breaking Bad there is this chemical, a compound known as methamphetamine, and for the synthesis of methamphetamine, the process which is used is reductive amination. So it is just the opposite of this process. So it would be a cool connection, but only it would make sense if you're a Breaking Bad fan and you pay attention to that series, which 99.8% chance is that you're not. So anyways, this is just another process then. But if you are one fan of the Breaking Bad series, then there is a cool little link to this process and the process which is used in the series, reductive amination. Anyways, we are talking about oxidative deamination. All right. Yeah. Okay. Oxidative deamination. What is that? You see, deamination. We talked about transamination before this. Now we are talking about deamination. I think these terms itself make sense, right? Trans means you're transferring. Amination means the amino group. So there is a transfer of this amino group, right? And deamination means you're removing that amino group completely. You're removing that. In what form are you removing that? Yes, ammonia. Remember what I said before? Ammonia is the form in which the nitrogen part of amino acids is got rid of. All right? All right. So what happens in oxidative deamination? Remember what we had formed in transamination, or rather what had been formed in transamination? That's right. Glutamate had been formed in transamination. Glutamine had been formed. Glutamate, not glutamine, glutamate. Now, listen very closely. This glutamate what would actually happen to this glutamate is it would be oxidatively deaminated to form its corresponding alpha keto acid again. Okay, so what's happening here? See, again this is reversible from glutamate by oxidative deamination. So oxidative means oxidation is also taking place. Oxidation is taking place. So what's happening? This is happening. This is the oxidized coenzyme NAD plus. This becomes reduced to NADH plus H plus. That is its reduced form. So if this becomes reduced, what is happening to the reaction? That is oxidation, right? Because reduction and oxidation are simultaneous. They go hand in hand. So NAD plus is getting reduced. So glutamate is getting oxidized, right? To what? To what we would say to be alpha ketoglutarate. Alpha ketoglutarate. Remember, we had formed glutamate from alpha ketoglutarate by transaminating that. But if we had transaminated, it means that alpha ketoglutarate had accepted amino group and had formed glutamate. So now, if alpha ketoglutarate is formed back again, what happened to the amino group? Yes, it is liberated as the golden boy ammonia. So ammonia is formed here. All right? All right. Obviously, this reaction would also require an enzyme. And what enzyme is this? This enzyme is glutamate dehydrogenase. Yes, glutamate dehydrogenase. This is a very, very important enzyme. All right? Right. Now, another thing which I would like to add here is that in this reaction, actually a water is also needed. A molecule of water is also needed in this reaction. Why it is needed, that is only kind of explained if you look at the molecular structures. I think in this case we can do molecular structure a little bit justice. All right, let's give it some justice. Let's look at the molecular structures here. All right, because glutamate is so common, glutamate and alpha ketoglutarate, these two pairs, these two molecules forming a pair, they are so common that we might as well know their molecular structure as well. All right, glutamate is a 5-carbon compound. All right, it is a 5-carbon compound. So let me first do that. 1 2 3 4 5 5-carbon compound. It is having, obviously, since glutamate is an amino acid, so it would have what? Obviously an amino group. It would obviously have an amino group somewhere. The amino group, suppose we say that this part is the N, nitrogen end. So the amino group is here, right? If the amino group is here in this end, this side becomes a C end, that is a carboxyl end. Alright, done. Now glutamate means glutamic acid means it's a acidic amino acid. So that means there has got to be another carboxyl group. There has got to be another acid group so that overall it becomes acidic. That is present here. That is present where? Here. Alright. Done. Done. Now this would actually be the molecular form of glutamic acid. But we are talking about glutamate. We are talking about glutamate here. So what is the difference between glutamate and glutamic acid? Very important to know. If I ask you what is the difference between acetic acid and acetate, what would your answer be? Very simple. Acetic acid is CH3COOH and acetate is an ion, so it is CH3COO minus. Same way, glutamic acid is this thing, so glutamate will be its ion form. Glutamate will be its ion form. Now the thing is that the amino acids mainly remain in their Zwitterion forms. So firstly, what we do is remove this and introduce the ion, an ion, that is a glutamate thing. Now we do justice to glutamate as a Zwitterion. Zwitterion means that the positive pole and the negative pole, they are coexisting. So glutamate is because glutamic acid loses a proton and forms a glutamate anion. And the Zwitterionic form of glutamate, which is more common to be found in the body physiology, that is NH3 plus and COO minus here also. So all the molecules, or rather all the poles, are charged, right? That is glutamate. All right, and obviously here we would have the corresponding hydrogen, right? This is glutamate. Alpha ketoglutarate will have the same 5 carbon part. All right, so 1 2 3 4 5, because there has been no removal of carbon dioxide, so same 5 carbon. All right, 5 carbon, we see that. But here what essentially would not be present is this amino group. This amino group would not be present in this carbon. What would replace this amino group? The replacement for this amino group will be the keto, that is double bond oxygen. That is why it is alpha keto, because this is a keto group. All right, so the replacement for the amino group is the keto group. All right, this remains as such, this remains as such, the rest also remains such. This becomes a keto group. This is the structure of alpha ketoglutarate. Now you say, if there is no water, what would the source of this oxygen be? Obviously, now you're getting my point why this water is needed. All right, the source of oxygen in this alpha keto group has got to be the water. That is why the C double bonded O, and that is why the water molecule is needed here. All right. So that is what we have in store with us. Plus, obviously, we would form the ammonia. All right. I hope I'm clear here. Now I said that this reaction was reversible. This reaction is reversible. There is a very strange thing going on. See, if this reaction is reversed, then what would happen? NADH plus H plus should become, after removal of these two reducing equivalents, NAD plus. In this reverse reaction, NADH plus H plus should turn into NAD plus, just the reverse of this thing. But no, that does not happen. In place of NADH, NADPH is used. NADPH is actually used. That forms NADP positive. So instead of NADH plus H plus, please do me this favor and remember that we form, or rather we use, NADPH in the reversed reaction. That is alpha ketoglutarate plus ammonia to glutamate. Alright. That is how oxidative deamination of glutamate takes place, and that is how ammonia is formed. This is a part of the metabolism of amino acids, right? Now, transamination is taking place in the cytosol of almost all cells. However, oxidative deamination is taking place not in the cytosol, but rather in the, you guessed it right, yes, mitochondria of the cells. So it is actually taking place mostly in the mitochondria. Okay. Again, there is a twist here. The twist is not in the part that it is actually taking place in mitochondria, but the twist is in the fact that, like, or rather unlike transamination, it is not taking place in most cells. It is taking place mostly in the liver. Mostly transamination, or rather oxidative deamination, not transamination, I'm sorry. Oxidative deamination is taking place in the liver. Liver cells, hepatocytes. Where in the hepatocytes? Mitochondria. Obviously, this does not account for 100% of the sites, because there are many other cells in the body also where oxidative deamination is taking place. But we can conclude by saying that mitochondria of the liver cells is the major site of again I'm mispronouncing it, oxidative deamination. Alright? Alright. So in this way, ammonia is formed in our body. This is an important landmark in the general metabolism of amino acids in our body. Why this is an important landmark? This is because the nitrogen containing part of the amino acid is not useful for us. Rather, it is toxic for us. It is highly toxic. That is why we remove it. We remove this ammonia which is formed from our body. How that takes place, I'll be talking about that in just a moment. Alright? Alright. So, what I talked about was how the nitrogen part was converted into ammonia. So what happens to the carbon skeleton of the amino acid? That is the next question that we should ask ourselves, right? What happens to the carbon skeleton? The most common answer is gluconeogenesis happens. The carbon skeleton is used for a array of various metabolic processes in our body, gluconeogenesis being one of the most major pathways where the carbon skeleton is used. What carbon skeleton am I talking about? Yes, the amino acid's carbon skeleton. And what happens to the nitrogen containing part of the amino acid? This happens. Ammonia. Ammonia happens. Ammonia, the source of ammonia in our body, major source we can say that it is transamination followed by oxidative deamination. So we can say that transamination and oxidative deamination are kind of coupled. They are coupled together. Alright? So there is a very beautiful sentences there about this coupling. That is, the two components of this oxidative deamination and transamination, they are not happening at the same site, right? Because transamination is taking place in the cytosol and oxidative deamination is taking place in the mitochondria of hepatocytes. So they are physically separated, but physiologically they are coupled. This is a very beautiful line, and this explains very accurately what is going on, right? Right. So the major source of ammonia in our body, I said that is oxidative deamination, right? But there are some other minor pathways producing ammonia in our body also. In fact, all, I repeat, all cells in our body form ammonia. So ammonia is kind of a waste product that is being formed, right? Now this ammonia, where was this formed? This was formed in the liver, mainly in the liver. But is 100% of the ammonia which is formed in our body formed in the liver? The answer is no. That is not the case. As I said, almost all cells produce ammonia by one way or the other because it's a metabolic waste product. So all cells are producing it. Protein degradation, metabolism, that is going on in all cells in the body. So ammonia is also being formed in almost all cells and tissues in the body. Now the thing is, what happens to this ammonia? This ammonia mostly, again, there is a variety of things that happen to this ammonia which is formed in our body, but mostly what happens is that the liver concentrates ammonia and pushes it into a cycle which is very famously known as urea cycle. Urea cycle forms, or rather converts, this ammonia into urea, and that urea is given out through the urine, right? That is the waste product, that is urea, in us, that is mammals. So we are said to be ureotelic. But before we go on to urea cycle, I think we will be covering urea cycle in the next video. We should talk about what happens to this ammonia formed, between what happens after the ammonia is formed and before the ammonia is pushed into the urea cycle. What happens in this interval? You see, I said that almost all cells form ammonia, right? But all cells in our body do not form urea, right? The function of forming urea is concentrated to which organ? I just said, yes, very good, liver. The liver is where urea cycle is going on. So the liver is where ammonia, which is formed in other sources, should be pushed at. Question is, how is the ammonia from different parts of the body pushed, or rather transported, to the liver? That is where we come to a different concept. It's a short thing, I won't take much of the time. That is trapping of ammonia. So we talk about trapping of ammonia. So ammonia is formed in different parts of the body, but ammonia is highly toxic. So the minutest quantities of ammonia are deadly to our nervous system. So we cannot, or rather our body cannot afford to keep ammonia. So it must be excreted out. To excrete out ammonia, first the thing that is required is trapping of ammonia, right? What does that mean? Ammonia as such cannot be transported. Like ammonia is, it's not like we dissolve ammonia in the blood and blood carries ammonia directly into the liver. It cannot be like that. What happens is, ammonia is actually trapped, or rather it reacts with glutamate, or rather glutamic acid. Glutamic acid, right? So what is happening? Let me tell that. You see, glutamic acid, I'm writing Ga, reacts with ammonia. All right. It is reacting glutamic acid here. It is reacting with what? What did I say? Ammonia, yes. And what is formed is glutamine. Yes. Glutamine is formed in this reaction. So glutamine is an amine. All right. Glutamic acid means COOH and amine means CONH2. All right. This step is an energy dependent step. So an ATP is needed here. ATP is needed here. Right. Also, another thing that is needed is water. Water. Right. The enzyme which is needed in this reaction is glutamine synthetase. So glutamine synthetase. All right. All right. So glutamic acid plus ammonia in the presence of water, ATP being utilized in the process, gives glutamine. Glutamine is what can be actually transported in the blood, right? So our blood, our body, in our blood, glutamine can be transported. All right. All right. Now we already know the structure of glutamic acid, right? We already kind of know it. So it is a 5-carbon compound and right, already 1 2 3 4 5, another minus. Actually glutamic acid, yeah, this is the thing. Sorry for being a bit confused. Yeah, this was there. From this, glutamine will be the same thing except that this thing becomes CONH2. This is amine. Amine. All right. All right. And water is actually, oh, did I say needed here? Oh, no, no, no. Water is actually given out. I'm very sorry. Water is actually given out. So please make a correction. Water is actually given out here. Why am I telling? Actually, why it struck me was because I wrote the molecular formula, the molecular structure, and that is when it struck me that water should be given out, not taken in. Why am I saying this? You see very carefully. You had taken in NH3, right? You had taken in NH3. So you remove a hydrogen from here, and since COO, let's suppose this has a H here. All right, let's suppose that this has COOH. So you take out the OH part, you take on the H from here, you take on the OH from here, then water should be given out. H plus OH, H2O given out. What remains is CO and H2. Obviously this is not the correct chemical explanation. Obviously there is entire mechanism and all, but as far as remembering this thing is concerned, I think this should suffice. All right. All right. So glutamine is formed here by the help of the enzyme, obviously glutamine synthetase, and this is taking place where exactly? This is actually taking place, glutamine synthetase. What did I say before this? That all cells are doing this. All cells are actually doing this. So glutamine is actually the transport form of ammonia in the blood, and so it is trapped as glutamine and transported to the liver. In the liver, we do not want glutamine, we want the ammonia back. So what happens is glutaminase enzyme acts. glutaminase enzyme acts on the glutamine here, and we get back the glutamic acid plus ammonia. Alright, and obviously since the water was given out, we need to include a water molecule here. This takes place in the liver. Alright, so we have successfully trapped ammonia in a transportable form, that is glutamine. We have transported the glutamine via blood to the liver. In the liver, the glutaminase enzyme is actually converting the glutamine back to glutamic acid and ammonia. Alright? Alright. Now, the thing is, glutamine is not the only transport form. Glutamine is not the only transport form. Just like glutamine, asparagine may also be a transport form. Asparagine. So glutamine was formed from glutamic acid. Asparagine will be formed from, very good, aspartic acid. So just like that, these are the transport forms, these are the trapped forms, these are the forms in which the ammonia is trapped and transported to the liver to be finally disposed of, as what exactly? Yes, urea. So that is where the urea cycle comes into play. Ammonia from all the body reaches the liver, then detoxified to urea by the liver cells via the urea cycle. And hence, we excrete urea, we being mammals excrete urea, and we are said to be ureotelic. Alright. However, there are obviously different classes in the animal kingdom which do not actually follow this urea disposal system. For example, fishes, they directly dispose of ammonia from their body in ammonia form itself. So fishes excrete ammonia itself, so they are actually known as ammonotelic. Ammonotelic or ammonotelic, I think. And AVs, class AVs and class Reptilia, they are uricotelic, that is uric acid excreting. We are ureotelic, that is urea secreting, not secreting, excreting, I'm sorry. So that's that. Alright, in the next video I think we would be talking about urea cycle. Alright, urea cycle itself is a very big thing, and we would be talking about urea cycle and its the disorders related to that, its regulation, the different enzymes related to urea cycle, and with that I think general amino acid metabolism would be close to the end. Then we would be delving into the specifics of amino acid metabolism. Alright, so goodbye. Thank you for watching. If you did watch, thank you.